nanohub.org.
Online simulation and more for nanotechnology.
So today's webinar the topic is DNA automation with nanostructured ceramics. This is going to be conducted by Dr. Mario Blanco. Mario Blanco is the CEO of a company in Tucson, Arizona called NANOPEC. So today's webinar is brought to you by NACK, the NACK Center. So the NACK Center is the Nanotechnology Application and Career Knowledge Resource Center, which is an NSF ATE regional center for nanofabrication education. NACK is a subsidiary of the CNEU, which is the Center for Nanotechnology Education and Utilization of Penn State University. And with that, I'm going to introduce myself again. I'm Zach Gray. I'm the Managing Director for Penn State's Center for Nanotechnology Education and Utilization. Vishal Saravade is our co-host, and our presenter today, again, is Dr. Mario Blanco.
Thank you so much, Zach. I'd like to, but first of all, thank you for this invitation, the NACK Center, Vishal, and Renee. It is a privilege for me to be directing the attention of our research and our directions in this webinar. And Penn State is very familiar to me. I lived in Pennsylvania for 6 years. That's where my two children were born, actually. If anyone knows where Abington is, Abington Hospital, I lived near Valley Forge when I used to work for a company that is now has been acquired called Roman Haas, very important company at the time. So I enjoyed those 6 years, except the very cold winters. I was used to California. So this is where I am now in Tucson, warmer weather. So I'm a graduate of UCLA, and I work at the California Institute of Technology for 16 years. I do have 65 plus publications, referee publications, and over 15 granted patents. And with my current company, I'm the CEO. Second time I am a CEO, and the fourth startup I'm involved in. So I'm very excited to tell you about our efforts to achieve the next level of DNA synthesis automation using our patented nanostructured ceramics. And I look forward to your questions. Before I get into that, I think it's important to understand why DNA synthesis automation is important. So our main goals include to synthesize genes that are difficult to clone. Often bacteria or mammalian cells have difficulty doing certain constructions such as palindromic sequences, simply because they have additional code in their genetics to prevent some dangerous constructs which will be useful or are useful in a pharmaceutical therapeutic setting. But also, we are very familiar with mRNA vaccines and therapeutic candidates for cancer, for example. And what we offer are to be able to make this without any animal products, without bovine serum albumin, or any bacterial E. coli fermentation, which is important because that may lead to contamination in the product. And even a small amount of bacterial protein may elicit an immune reaction during clinical trials. And although you may have an amazing new therapeutic, the FDA might not approve it because the 1%, 2%, X% of your population had an immune reaction to the residual protein, bacterial protein. So, and it's very expensive and limits the number of tests that you successfully can do if you're all the time worrying about this type of contamination. Also, to provide large-scale gene synthesis, it's important for two areas. One is DNA banking. It so happens that NANOPEC provides materials, chips, for the largest DNA bank in the world. And we're also looking at archiving. You may have or may not have heard, but DNA is a long-lasting medium to store information. We know that because DNA from Neanderthal 40,000 years ago has been decoded. I think this year's Nobel Prize went to the scientist for medicine and physiology responsible for decoding the Neanderthal genome. So DNA lasts a lot longer than your DVDs and CDs and even your flash drives. It's estimated in the tens of thousands of years. If you keep it under, let's say, encapsulated inside a steel bullet or capsule and filled with argon, it probably supersedes 40,000 years. So companies like banks and, of course, iCloud companies that want to archive data, they're looking at DNA as the means to do that. Also, we want to optimize codon use to boost targeted protein expression, meaning you want the messenger RNA to be as fast as possible to go through the ribosome to express more protein faster. And so because there is redundancies, you may want to try multiple variants of your DNA, which gets translated into messenger RNA for translation. Another area of interest is CRISPR. Of course, you have heard, you probably know very well of the already few successes with CRISPR-Cas9 edited genes. There's been some recent successes in terms of full remission or cure of certain conditions. So, of course, that is a very sophisticated biological tool, but sooner or later you need to include the gene that you want to add using this technology, and that means you need to create that gene de novo. So I want from the onset to say that there is a difference between using the biological means to replicate DNA. That's not what we're talking about. We are talking about creating whole new sequences, if you will, single-stranded DNA, if that helps you understand what we're doing from scratch. So you just type a sequence in a computer, and then the synthesizer, the materials, the chemistry does the rest. So we will hopefully be of use to CRISPR-Cas9, CRISPR companies that are doing gene editing. And of course, recombinant antibodies are very important. So how can we humanize them to create them in good amounts in let's say mammalian cells, which may or may not be human cells. So this is just a number of areas that DNA synthesis automation can really improve. And what we're talking about is not a factor of 2, it's not a factor of 10. We're looking at factors of 100 in terms of speed and efficiency and accuracy or above. So that's the goal. In short, we want to realize the full potential of synthetic biology, which we always say is to protect, meaning vaccines, to heal, means therapeutics, but also to improve the quality of our lives. Some of us say longevity or extend life, but life needs to be extended with good quality. We don't want to live for a very long time with bad health, right? So I'll talk to you about the fundamentals, the applications. I will make a stop for questions from you. Please, in the middle of the presentation, I'd like to hear some of your questions. We have plenty of time. I speak fast. I hope you understand my accent. And then we can move forward to the last part of the presentation on a little bit of how this works and what is the next level of automation that we have accomplished, and we are also continuing our efforts to increase. But before getting into the subject matter, I'd like to review for some of you who may not be physical scientists, like chemists or physicists, or material scientists, or engineers. What is a nanometer? So a nanometer is 10 to the minus 9 meters. So that's 0 point followed by 8 zeros, then a 1. That's the size of 1 nanometer. But for us, this number, this dimension is not within our daily experience. So I'd like to show the following. Apologize, this is for high schoolers, but it's been very effective in teaching what a nanometer is. And we will mention this number quite a bit. So this is a scanning electron micrograph of a human hair. The width is what I want you to pay attention to, not the length. And that's about 80,000 to 100,000, varies from individual to individual, but that's 80,000 nanometers in width. And this picture here at the bottom is, of course, is the eye of a needle. But if you squint or look very carefully, there is a small, tiny figure at the bottom of the eye of this needle. That is a sculpture. That sculpture was done at the University of Vienna, and it was done using laser interferometry and curable resins. And of course, I'm going to show it to you. But what I like to do is show that sculpture as if it was positioned on the surface of that hair. So roughly that sculpture is about 10,000 nanometers in height. And I'm going to continue this exercise and then show you just the arm. So the arm is 2,000 nanometers in length. And now just the hand is about 500 nanometers. And now I'm going to expand only the thumb. And once we're here, that's where I want you to be, because that circle over there, 50 nanometers, that is a typical size of the nanostructures on our ceramics that we're able to fabricate. And so this begins to give you an idea for how small the structures, the pores on our ceramic films are. Don't worry, I'll stop for questions in a little bit. But why is the small important? Well, small means fast. I mean, if you look at the rate of movement for insects and then bacteria, you're talking about, not seconds, you're talking about milliseconds. And the smaller any feature, the higher the frequencies, the faster the movement. So the idea then that we had was, can we apply the same concepts that made electronic chips fast into the gigahertz? Potentially, we will reach levels of that, but the idea is then reduce the size of your working elements so you can attain high frequencies. And so let me go back to 1947. When the transistor was invented. This is the first transistor. It does have the 3 electrodes as any ordinary transistor, and it was a big crystal. The size of this is 1 inch. So keep that in mind, because for the current workhorse of DNA synthesis, we're talking about 1 inch as well. But 1 transistor means we can store 1 bit of information. A 0 or a 1. But in 2020, Apple came out with the A14 chip, which is a quarter of an inch in length. And you have 11.8 billion transistors. And the speed is also amazing. You can actually upload or store 4 full human genomes per second. That's how fast that chip runs. So small reduction in size equates with higher speeds. This is the current workhorse for DNA synthesis. These are either wells in a microplate or, as it's shown here, a column, a plastic column, and it's filled inside with what is called a frit. This frit is made of compressed glass beads. And they named this control pore glass, invented in 1934. And the largest number of oligos you can make is using the most refined or the high density microplates with 726 oligos. And you can see the growth of this has been linear, and it's taken a couple of decades to get from 24 to 726 oligos. But the good thing about it is that in a single column, you get 1 micromole of DNA. So just to remind you, an oligo is a piece of DNA. It may be typically single-stranded, and roughly today, The standard is 60 nucleotides, 60 bases, but there are companies that do 100, some that dare to do 300. This is one of our products. It's a chip, 1/16 of an inch, so it's much smaller than the Apple chip. And in the center, there is a optical code. This is like a QR code. Think of it as a QR code. It's actually data matrix code, but it's very similar. And our customers use this to direct that chip through a chemical flow chart. So they know when to add A, C, T, or G to that particular chip. So one oligo per chip. And we've been making and selling this since 2021. But we started the work in 2019, at the end of 2019, pre-pandemic. So it came out really handy when there was a need to speed up DNA synthesis. In that single chip, you can make also a single oligo, and then one of our customers can do overnight 32,000 chips. So basically, that allows you to do multiple variants of a large gene overnight. We are now moving to a plate, which is 10 by 10 centimeters. That's for our internal synthesizer. This is what our customers use, but we are going to a different format. We want very large chips where we can do 180,000 oligos maximum. And the nice thing is that the scale of this chip will allow us to do 1.8 micromoles, which is very similar to what can be accomplished with standard low automation methods such as control pore glass. How do we do the chemistry? Well, it's pure chemistry, right? So there is no enzymes involved, there is no bacteria, there is no fermentation involved. So there is this cycle is accredited to Cariters. Cariters came up with this in the 1960s, I believe, and it's a multi-step process. Each cycle basically adds one base to the growing oligo, and it tries to avoid errors by doing certain operations such as capping, for example, where you stop any DNA chain that has not added the current base. So if you do this carefully, you can achieve high quality DNA, single-stranded, and also in good yields. So up to now, we have applied a very simplified workflow. We have been providing a platform for our customers, which means we provide the ceramic film or chips. In addition, we have proprietary technology to get the first base placed on the chip in a way in which they are spaced out, but not too far away from each other. Think of it as growing a forest. If you put the seedlings far apart from each other, each tree will grow very well because there won't be shade from other trees around it, but your overall yield will be low. If you go the other way, if you put the seedlings too close to each other, your yield will also suffer because you will have lots of trees, but they will not grow as well. So there is a Goldilocks medium, I guess, we can call it. And we have the technology, proprietary technology to do that, and also to verify that, yes, the distance is the appropriate distance. So this is what we've been doing, but now when we send this to our customers, then they use their own synthesizer to grow the gene of interest to them. But many of our customers don't have that expertise. That's the reason why we're moving that all the way to the right, where we don't do the DNA, but we will provide a turnkey solution to our customers, both commercial, academic, so that they can do this themselves without disclosing their precious sequences to us. Typically, most companies use outsourcing, meaning they go to a company outside. Examples in the US include IDT, GenScript, Twist. Outside the United States is much cheaper, so you can go to, for example, Wuxi, which probably makes more DNA than any other company for pharmaceutical therapeutic use than any other company in the world. So if you don't send those sequences outside the company, then no one knows exactly how you are doing what you're doing. Even though you may have nicely signed documents, you don't want to risk those sequences to be, at least during the R&D and the preclinical tests, to be known by others. Now, what is the core of our technology? I call this DNA paper. It's not paper, but I believe that attempts to print DNA as opposed to micropipette with high densities and frequencies have been done. There are patents going back to 2004 where you will find DNA printers using, at the time, the highest resolution available, below 300 dpi, dots per inch. But since then, even a house printer, can do very high resolution, 1200 dpi and above. So it's been tried, and it was tried on paper, it was tried on glass, and there are issues with those materials. On paper, you can have bleaching of reagents from one well to another, from one piece to another, and that means contamination. On glass, you can avoid that by only activating pieces of the glass surface, small regions, but it's very hard to activate glass, and given that you only have the flat surface to work with, your yields are going to be on the order of picomolar per square centimeter. So there is a commercial printer you can actually buy, but it's always sold for academic purposes. It doesn't have the power to generate sufficient amounts of DNA or RNA. By the way, the chemistry can be directly applied to produce RNA, not just DNA. So it is a research tool. It's not something that can just, academic research, not something that can be put into a commercial environment. Maybe you can, but you need to do PCR. You need to do amplification after that. So what we have is this ceramic. It's nanostructure, meaning it has pores and they are not random. They are truly controlled pore size. We can truly call this controlled pore size pores. We control the diameter of the pore as well as the length of the pore. As I mentioned, we also have the ability to control the surface chemistry, the surface spacing. And we have patented this platform. We call it DNAReax. So this is a scanning electron micrograph. I admit this is one of the best materials that we produce because we can produce them order like a honeycomb system here, where by changing the conditions, typically working at low temperatures, meaning low speeds of fabrication, we can create highly ordered nanostructured ceramic films. But it doesn't really confer you much of an advantage over a disorder or quasi-order ceramic film. As long as the pores are all vertical and there's no crossovers, no contamination, we can use a slight disorder ceramic films, and they are faster to produce. So we don't typically aim at getting order like this to create the lattices, such honeycombs. Hexagonal close pack is the official name of this. But some disorder, is admittedly employed so that we produce this material much faster. So this is market-ready technology. It's been customer tested already with high performance. They report data to us. It's not our own analysis, it's their own data. And we work really hard to lower the cost and do scalable in-house production of this ceramic film. And we patented this DNA automation platform. One of the interesting parts of lowering the cost was actually, I would say, incidental to the desire to lower the cost. And that is that academics and other companies use toxic chemicals to produce similar materials. They include mercurous chloride as well as hexavalent chromium. And I decided I wasn't going to use those chemicals around my lab and expose my staff to this. So we found ways of doing this process without using mercurous chloride or hexavalent chromium. And that translated into savings because we don't have to pay hazmat, transport, protective equipment, PPE, and also hazmat waste disposal of contaminated solutions. So that desire to protect our staff translated into significant savings. We estimate about 75% savings in cost. I want to just briefly indicate that most of the reaction, 99% of the reaction, occurs inside the pores. And I'm showing here an schematic view of the pore and that square top right C square. And then I kind of blow up the inside of the pore, and I kind of show an RNA, in this case, an RNA chain growing inside from the walls. So this is solid state synthesis. The material is held covalently bound to the interior of the pore while it's being grown one nucleotide at a time. And once you're done with this, then you cleave it from the surface. On the top left, you see the actual structure of control pore glass. So I don't know why it's called control pore, because it looks to me like coral, but it's beautiful though. However, the pore size distribution is very broad, not uniform, and that leads to a problem where if you focus on small pores, and we specialize on sizes between 50 to 400 nanometers in diameter, the smaller the pore, the more DNA, the more RNA you're going to grow, but there is limits to how big that oligo can grow, because eventually it's too big to fit inside the pore. So this material, control pore glass on the bottom left corner, has pores with very small diameters, and that leads to something technically called shortmers, which means the oligos are not the right size, and you need to cap them so they don't waste more of your reagent, and that means lower yields. So the strategy to overcome that is to go to very large pores, typically 1,000 angstroms and above, much larger than that. And that means lower yields, because again, it's like, growing a forest. Okay, so I'm going to stop here.
Yeah, absolutely. So, so we have one here. Can you please elaborate on the quality assessment protocols of the DNA or pore nanostructures that are prepared?
Yes, very good question. As I mentioned earlier, our customers are the ones that are providing that information to us, but we have already reached out to commercial and academic labs so that we also will get our own internal validation. So typically, just to confirm the size of the oligo, you can do LCMS, but you actually need to do the sequencing. And our aim is to provide the yields of the desired sequence, and then the amount of single, double, triple base nucleotide mismatches. Because one of the things that we are pioneering is we don't want to use any polymerase. And that means when we synthesize, given our ability to automate the process at very high speeds, and I'll talk a little bit about that in a moment, there is no need to do partial matching of oligos from the 3' to the 5' double helix. We can actually fill both sides. So at the end, we are left with all bases in place, and then the ligation is the only step that requires an enzyme to be able to covalently link those oligonucleotides. But we decided not to use polymerase because there's some errors in including the right base. There is mismatches. Of course, nature produces amazing enzymes that have been evolved in the lab, that can do that more accurately than ever. So I'm not saying that those have high error rates. I'm saying that we don't take the risk and we generate the full double-stranded, full complementary. So we are going in that direction to do full sequencing. It's cheaper, actually. I found that out, a lot cheaper to send a ligated 3,000 base per gene for sequencing than it is to send the corresponding 100 oligos. So we will get the information that our customers want because most of them, not all, are interested in genes. Not interested in individual oligos. Some companies do, like companies that do probes for imaging or vaccines or microarrays. Yes, they want oligos. And I think there is a drive to go to longer oligos for those purposes. But we are very narrowly focused on the ability to do synthetic biology. Later on, we will pay more attention to genomic diagnostics. I hope that answers your question.
Yeah, excellent. Thanks, Mario. And there was another question about the geometry of your nanostructures. So would you be willing to talk a little bit about how the ratio of the pore depth to width impacts the rinsing during synthesis? It seems like that might be difficult in the format you're using, but it sounds like you might have a solution for it.
Yes, excellent question. You're not the only one who asked that question. We've had conversations with very well-known scientists around the world, and that's one of the first questions that comes out. I'm hoping to be able to make an announcement within a couple of months at the most on a major company led by a very well-known scientist, and that was one of the questions. And what better manner to answer that question than sending a video? So the question was, as a scientist, he said, I know these are capillary, and capillaries have very strong forces. So if you don't put vacuum or if you don't blow, let's say, noble gas like argon, you're going to have a hard time making your reagents go through these pores. Well, I knew already from my customers' data that was not the case, but I wanted to understand that. We wanted to understand that better in our lab. So about a couple of years ago, we started a very simple experiment, which is we take our material, our ceramic film. Typically, we like to do 75 to 100 microns in thickness. So if you look at 100 micron pore, we are looking at shape factors of 1 to 1,000. So the pores are 1,000 times longer than they are in diameter. So we began testing with water. We found the nanostructured ceramic. By the way, NANOPEC means nanostructure performance enhanced ceramics. That's what the name means. So we modified the surface to make it either more hydrophilic, more hydrophobic. We have control over that chemistry. And so we found this material to be anisotropic. And we understand now why. I can't tell you, but water on a 100 micron film takes about 15 to 30 seconds to transverse without at atmospheric conditions from one side of the ceramic film to the other. But if you flip it, it takes 1 to 3 minutes. So it's anisotropic, and I think we now understand why. However, water is not the medium utilized to do this synthesis. The solvent use actually has to be very dry, no water. So the solvent used for this is actually no water. And using that solvent, it takes 5 seconds because it's very low surface tension. So in 5 seconds, the reagents go through the entire fan. All right, so let's move on. All right, so I mentioned one of the world's largest gene banks. Actually, it is the largest, is using DNAReax. Their synthesizers, you see here a picture of their lab. Each one handles 8,000 chips, divided in 4. That's 2,000 chips. And then the chips have this code on the surface that I mentioned that we create, so that there is a sorter inside, and then they patented this technology, and they send each chip to the correct next reaction bath, and that's how they can make 200 gene variants overnight. And when I talk about genes, we use as the standard the spike protein gene, so 3,000 base pairs. So they are able to make 200 variants of this gene overnight with one of their synthesizers. Unfortunately, no one in the United States has this mindset. I'll explain our own mindset in the US is different, because what they're able to do with this technology now that we replace their silicon chips, they were using silicon chips, with our chips. Initially, they called me up after the first test and they said, "Oh, Mario, we're getting already 75 times more yield." And I replied, "Only 75? I expected more." And they laugh on the other side because, of course, they don't have 75 synthesizers. So just moving one of their synthesizers to our material, they should be able to produce more DNA than in the rest of their entire synthesizer floor. But over the last couple of years, we've continued the effort to improve the yield, and now the yields are 460 times. By the way, we made it possible to make no changes in the synthesizer. So we took it upon ourselves to make a chip that fits inside the synthesizer, and it has all the necessary technology to carry the process as they were used to doing it. Good. So here I have a performance comparison between CPG, control pore glass, which is the traditional method, most widely used in the United States. Second one is silicon, which is used by companies like GenScript and Twist. Twist, I think exclusively, I may not be right about this, but they exclusively use silicon. And GenScript uses both CPG and silicon chips. But what is important are these circles. There's a lot of data here, but I just want to say we have achieved coupling efficiencies, which is the probability that you add the right base, given that you added n bases. What is the probability of correctly adding the n+1? And our customer is reporting greater than 99%. The industry standard these days is 99.5. In some cases, we have reached 99.7%, and we are trying to reach 99.9%, which I believe has not been achieved. I mentioned we are the only substrate. Remember, this is all solid state synthesis, and we are the only one that has this high precision chemistry method to space out the DNA as they grow on the surface. Another aspect of our material is that the first step, the so-called chemical initiation, is very, very short. We're talking about less than an hour, and using non-toxic materials available in your local pharmacy. Whereas to initiate silicon CPE, to activate it so that you can do DNA growth, requires overnight treatment with piranha solution. I mean, just the name, I stay away from something called piranha. So these are includes hydrofluoric acid and nitric acid, sulfuric acid mixture. So it's very corrosive. And at the end, they don't achieve the same levels of activation of the surface as we are able to achieve in less than an hour with less toxic materials. So now, We haven't tried going beyond 60 nucleotides, but I don't see a reason. We've done some estimates in terms of computational simulations about the size of the oligo we can fit, and we can go to 120, perhaps 200. Just making the pore larger is the trick to allow enough room for reagents to go and diffuse through our material. One last comment is that, as I mentioned, for us, automation is to create as many oligos as possible. So silicon already claim that you can do 1 million oligos in one chip. However, they can only do it at femtomolar amounts, 10 to the minus 13. Whereas we can go anywhere from nanomolar, which is enough to do preclinical trials, to micromolar, which is enough to do clinical trials. So we have the ability to print in regions of our chip to increase the yield. So there is additional control. Thank you for that. All right, so let's see what else. Our current focus is on early adapters, companies, academics who want to try this out. For applications in biological therapeutics. So DNA banking is basically done. We're happy with where we got with that. DNA-porous may be in the future, but right now our focus is vaccines, DNA, RNA-based therapeutics. Now, we don't make them. What we are focused on is enabling our customers to do that in-house. So there's a lot of hand-holding, a lot of resistance sometimes to break the habit of sending your sequence, precious sequence, to other countries and risking losing your intellectual property. So hopefully, by making it very simple, easy to use, turnkey solutions, and they might be able to save themselves those issues. But there are more important, I believe, other than intellectual property protection, aspects to doing synthetic genes. And that is that we're able to create preclinical amounts of cell-free, animal-free DNA overnight. That means no bovine serum, which is used for mammalian cells, or E. coli, use of E. coli fermentation, which is the traditional means to do plasmid amplification. There's no need for plasmids. So therefore, there is no risk for cell DNA and protein contaminants that can lead to undesired immunological reactions in preclinical trials. And we have already sufficient yields to run preclinical and clinical tests directly from synthesis, no fermentation or amplification needed. So we are at the center of those three circles in terms of good yields, cell-free and animal-free. I should say, though, that there are constructs, as I mentioned earlier, that are not possible to do through fermentation because E. coli has a mind of itself. It has its own code that says, oh, I refuse to make that sequence. It's not good for me. So there are cases, I won't go into details, but one of our customers is coming to us so we can do those constructs for them. So we're collaborating with pharma, developing DNA-based therapeutics. Of course, once you have DNA, you can make RNA through reverse transcriptase. And so, or we can actually make it directly using synthetic bases for nucleotides for RNA. No major change. So the supply chain and the timing is the number one issue for progress. It takes 2 months or longer to get a single gene in preclinical amounts, sending out your sequence. But it's also expensive. And so the standard lead times for external suppliers is 6 to 8 weeks for a single gene. Whereas we are producing sufficient yields for required gene libraries and animal tests that can be created on our new format, 10 by 10 centimeters in a single day. I think I mentioned already this, so I'm going to skip, but it's a little bit of the chemistry that goes on into creating the first covalently bound species on our ceramic film. This is common to controlled pore glass. There is nothing proprietary here. What is proprietary is how do we achieve that uniform coverage and control the spaces between those molecules so that you can grow DNA well. So we have 6 patents, some of them granted and some of them applied for worldwide. So all our inventions, we have a strategy. We patent everything that can be reverse engineered, and believe me, there's a lot of that going on. And anything that cannot be reverse engineered, we just keep it as a trade secret. So that's our strategy. Good. Well, thank you so much. I'm going to turn this back over to Zach and maybe entertain some questions now.
Yeah, perfect. Thanks, Mario. Excellent webinar, super interesting. So we do have one more question. All right, it says, who are the typical customers, like typical industries for DNA chips and pore nanostructures?
Oh, who are they?
Yeah.
Well, DNA banking was there. I mean, DNA banking, they are early adapters. Any biological pharmaceutical company, biologicals are anything from antibodies, proteins, peptides, RNA, DNA that have therapeutic uses. Those are our customers, because even if they produce antibodies, they need to generate gene libraries, DNA libraries that work with the area of the antibody that is responsible, the variable region, which is responsible for the specificity of the antibody. But also, we are hoping to catch on some of the gene replacement therapies, CRISPR, Cas9 companies. We're looking for early adapters. The customers that we have considered or are very easy to approach and immediately come on board are early adapters, are companies that they have tried everything. They have tried the standard methods and they can't get what they need, or they get it, but it takes a very long time and it's costly. So those early adapters it's very sweet to work with them because they have a need and we have a solution. And rather than rather than spending money and time doing marketing, we prefer to work one at a time with customers and then keep them as lifetime customers. So we are not here to obtain 5,000 customers in a year or something like that. No, our growth strategy is to be a tiny little BASF. We don't make the final product. We make the final product they make better. And that means a lot of technical support and technology development for them, very tight research-based collaboration. And that means we are also very open to working with academia. So if anyone is interested in a specific construct and you would like to co-write either an STTR or your own NIH grant and mention us as a supplier, we'll be happy to collaborate.
Awesome, thanks. Yeah, I'm sure that we're recording this, and we'll certainly the word will be spread of what you're doing. So hopefully you can find some good collaboration. But this concludes our webinar. We'd like to thank Mario once again for taking the time to put this together and to speak with all of you, and his very nice, diligent responses to your questions. So thank you all. And so Mario, great feedback from the audience so far. So thank you very much.
Thank you. Bye-bye.
Bye-bye, everybody.